I would like to welcome everybody to the ISAPP webinar on the human microbiome. And joining us today are several really distinguished speakers, and we're going to be introducing them shortly to go over the different topics we're going to be covering. And I would like to start out by introducing my co-chair, Eamonn Quigley, who will give a few introductory comments about the webinar.
Okay, Marianne, can you hear me?
Yep.
Okay, welcome everybody. And the first question everybody's going to ask today is, what in the hell is Quigley doing in a symposium about the microbiome? Well, I'll give you a slight little bit of background because I want to say this to address it to all of the speakers. So for me as a clinical gastroenterologist, the microbiome may seem like a distant entity, but in fact, it's a clinical reality. And on a virtually daily basis, and I'll talk about this in my presentation, we are asked by patients, do they have candida? Or do they have some other fungal overgrowth in their gastrointestinal tract? And my interest in the symposium today is to hear about where we are with techniques to evaluate the human microbiome and where we are in terms of understanding what role it has in health and if it has a role in disease other than in extreme circumstances. So for me, this is a very personal area of interest, and I think it's an area of great interest and importance to many practicing clinicians because of all the stuff that's out there in the internet about fungi and their role in supposed role in a whole variety of diseases. So I'm delighted to have the opportunity to be with you today. I apologize if I suddenly disappear. This is actually the worst day of the year for us. It's the 1st of July. We have a whole batch of new residents and fellows, and we've added chaos to bedlam. So please excuse me if I disappear transiently. And now I'll hand back to Mary Ellen, who'll give you some housekeeping and give you the rules of the game. Mary Ellen.
Thank you so much, Eamonn. I want to also add, for those of you who are not familiar with ISAPP, ISAPP is a nonprofit organization that's dedicated to advancing the science of probiotics, prebiotics, and related substances. And I encourage you to check out our website, which is isappscience.org, where there are many resources on publications, blogs, infographics, and videos. And you can also sign up for our monthly newsletter on our website, and that will make sure that you don't miss any other types of programs or webinars that we might offer that you might be interested in. So a couple of housekeeping items I want to go over. The session will be recorded, and most of the content will be posted on the ISAPP website. So not all the talks, but most of the talks will be recorded. And so it'll take us a couple of days to get that up, but if you happen to miss the live broadcast, you can see the recording. And because our schedule is so packed today, I'm just going to introduce speakers, or Eamonn and I will introduce speakers just using their names and affiliations, but you did have links in to their bios in your the information you received on the webinar, so you can look up all the details about each of our speakers. Also, please submit your questions using the Q&A function. So all questions will be submitted in a written form, and then at the end we will have a 20-minute Q&A panel where we can go over them. So I look forward to that. But so to start out, we're going to have 4 roughly 20-minute presentations that are going to cover the basic science of the microbiome. And then we're going to have 2 shorter talks that are going to provide provide different perspectives, one from a clinical point of view and one from an industry point of view. So let me hand this back to you, Eamonn, to introduce the first speaker.
Thank you very much, Mary Ellen. One of the other reasons I want to be involved in this was the opportunity to introduce somebody whom I'm very familiar with, Pauline Scanlan, who comes from APC Microbiome Ireland, excuse me, and the School of Microbiology at University College Cork, which for many years I had a very happy affiliation with both of those. So Pauline has done quite a lot of work on the microbiome, including on techniques to evaluate the microbiome. So it gives me great pleasure to invite her today to kick us off. So Pauline, it's all yours.
Okay, thank you very much for the kind introduction, Eamonn. I just want to check everyone can see me and hear me. Yeah, okay, great. Thank you. So I'll just share my screen there. One moment to get started. Here we go. Okay, so again, thank you very much for the kind introduction and good afternoon from a very sunny and warm Cork. As Eamonn pointed out, I do quite a bit of work on the gut microbiota. I'm a microbial ecologist by training, so this is the perspective I come from. And my brief today is to provide you with a very general introduction into the human gut microbiota. And so just to give you an overview of my talk, as I said, I will give you a very general introduction to the human gut microbiota. So by necessity, my talk is kind of general. It's a little bit superficial in places, but there's a couple of key points that I would like to make that I hope people can take home with them. So as a microbial ecologist, I'm interested in two things. So fungal diversity, so who is in there in terms of different taxa, species, genre, and so on. But I guess the key question in microbiome science generally is the functionality of the microbiota and the microbiome in this case. So what are these fungal species doing in the human gut? So what's their interaction with their host, their environment, and how do they interact with other species that are present in the human gut? So before we move on to the main of the talk, of course, and Eamonn has alluded to this, that the fungal community of the human gut microbiota, and it's been largely ignored in the last few years, but it is a very, it is a reality, a clinical reality, and it's an area that's receiving a lot of research attention recently, thankfully. And I guess one of the reasons that it has been ignored is, I guess, the perceived low numbers that members of the gut microbiota have in the human gut. So if we actually look at the broadest level of taxonomic classification, fungi are present at relatively low numbers when you compare them to the bacteria. So if you do a metagenomic sequence analysis, typically you will find that less than 0.1% of the sequence data can be attributed to eukaryotic or fungal origin. So if you look at the individual species level, however, single species of fungi, they do occur in the same range as single species of bacteria. So for example, species like Candida can actually comprise up to 10 to the 6 cells per gram of feces. So the key point here is that although the microbiota or the fungal community as a collective at the broadest taxonomic levels is present in relatively low numbers to the bacterial fraction of the community, individual species of fungi do occur in the same numbers as certain probiotics and pathogens of interest do. Another point I'd like to make as well, that microbes at low numbers can have an effect. So you don't have to predominate a community to have a physiological effect on hosts or to have an effect from a community perspective. So we know in microbial ecology that keystone species, so these are species that play really important roles in communities, these can often be present at very low numbers. And another point I think that's really important to make is that fungi are fundamentally different to bacteria. So in many respects, microscopic fungi, the only thing they have in common with bacteria is the fact that they can be seen under a microscope. However, they're fundamentally different in terms of cell size, surface area, volume, and the genetic content, of course, is much different than bacteria. So they're fundamentally different to bacteria. So what the analogies or the comparisons we can make between fungi and bacteria and their roles in the human gut, can be quite limited. And another point I'd just like to make, and this is just a more general point about fungi, is that they're not just ignored in terms of gut microbiota research, they're ignored generally. And this is a lovely editorial that appeared in Nature some years ago. And it's kind of a call to arms to stop neglecting fungi. So although many people perceive fungi to cause harmless infections, actually 1.6 million people die annually due to fungal infections. And as well as that, and this is quite a staggering statistic, 1/3 of global crops are destroyed annually due to fungal or oomycete infection. So moving on then to the main of the talk. So I guess the key question is, why are we here and why are we talking about studying the human gut microbiota? So as we know, microbial ecosystems like the human gut, they're extremely complex and their functionality is often greater than the sum of its parts. However, to date, we've just primarily focused on the bacterial fraction of the community. And this has resulted in a very one-dimensional view of the human gut microbiota. So it's very clear that we need to start studying fungi in more detail, and this is required to better understand their ecological and functional role, both at the species and at the community level. So we need to identify their roles in ecosystem assembly, structure and function, and we need to identify any potential fungal pathogens, commensals or potential fungal probiotics that can act alone or interact with others to either help or harm the host. And of course, all this information is hugely important, of course, because I guess the holy grail of microbiota research is to beneficially manipulate the microbiota, to benefit the host. So we really need to know who is in there and what they do, and not only what they do generally, but what they do in different host genetic backgrounds. And this is relevant, of course, to fecal transplantation, the use of fungi as probiotics, and of course, kind of personalized or individualized medicine. I'm going to start off now, and I'm just going to get into more detail on diversity. So the diversity of fungal species present in the human gut. But before I get into the detail, I guess the key point that Eamonn alluded to is like, how do we actually go about studying the human gut microbiota? So what tools are out there, or how are these studies conducted in the first place? So just to give, just to start off with, you typically take a fecal sample, or you could take another sample of interest. So you can do a culture independent analysis. So this revolves around doing a DNA extraction that's robust to extract the fungal component of the community. The gold standard in terms of the gene to target is the internally transcribed spacer. So this is a region of the fungal ribosome. So you usually use primers that are specific to ITS1 or ITS2. These are amplified using PCR, and then you can do next generation sequencing. Another potential approach that isn't widely used for many reasons, that due to obviously the low relative abundance of fungi, is you could go straight from DNA extraction to shotgun metagenomics. But you would have to sequence at an extraordinarily high depth to actually get any useful information. So this is not widely done, and there is very little available in the literature on this. Using this culture-independent approach, of course, we can characterize taxonomic diversity within and between groups of interest, and also look at fungal communities over time. And we can identify potential functional diversity between groups. The second approach, and these can be used in combination, is to do a culture-dependent analysis. So this is what would have been traditionally done, and it's also continued to be done in labs at the moment, which is great. So you take, you plate your sample on fungal selected media. So there's a whole range of different fungal media available. You can select isolates then for genetic and functional analysis. So you can do whole genome sequencing. You can do host microbe interactions using in vivo models, and of course you can do a whole array of functional screening and functional analysis. So using this approach, you can characterize functionality of specific taxa at high levels of genetic resolution. You can look at host microbe interactions, and of course you can get a really good sense of interkingdom interactions. So you could look, for example, at interactions between specific bacteria and fungal species. So of course there's pros and cons associated to all these methods. There's issues with databases for fungal ID, and there is actually no gold standard on consensus on best practice. But this is a general overview of what kind of methods are commonly used in these studies. So to move on then, so what do we actually know based on these types of analysis about the diversity of the human gut, microbiota? So in 2017, we did a review of the available literature on the fungal fraction of the human gut microbiota. So we evaluated about 40 different studies. And I'm just glossing over this, as I know the other speakers will get into these variables in more detail later on. But the gut microbiota, it clearly varies according to methodology, to age, diet, and disease status. At the phylum level, the vast majority of fungi associated with the human gut belong to the Ascomycota or the Basidiomycota, and that's irrespective of age, diet status, disease status, and so on. Within these phyla, a diversity of different genera and species have been found. However, and these are two important points I would like to make, Much of the diversity reported in the literature, so in this analysis we did, was study specific and often specific to an individual person. So much of the diversity is just exclusive to one study. So a lot of the diversity isn't common to groups of individuals. And so just to make this point a little bit clearer, so what you can see here are the main phyla associated with the human gut microbiota. And on the y-axis, you have the number of different genera reported for each of the fungal phyla. So you can see, as I said, the vast majority of fungi are associated with Ascomycota and Basidiomycota. However, if we remove the singletons in the dataset, so if we remove a specific genre that are only reported once in a study, you can actually see the dataset is greatly reduced and that there's very few genera that are common to all studies. However, even though a lot are study specific, you do actually find specific genera that are more widely distributed. And of course, these are Candida, Saccharomyces, Malassezia, Penicillium, and Aspergillus. So I'm going to talk about these 2 points in a little more detail in the coming slides. Another point I would like to make, however, is that the gut microbiota is characterized by low evenness, which means that communities are typically dominated by one or two genera, and the vast majority of diversity reported is present at a very low relative abundance. So to illustrate these points, I'm just showing you data from a 2017 study we did. So you can see on the y-axis the relative abundance. So the details of the different codes are not actually that relevant, but I will talk you through the graph in one minute, just as we go through it now. So this was a study of 18 healthy adults, and the graph, as I said, shows relative abundance of different fungal genera based on culture-independent fungal ITS analysis. In the entire data set, so from the 18 individuals, we got 181 different fungal genera. However, all bar 9 genera were present at less than 1%. So what this means is the vast majority of diversity that we uncovered in our data set was actually present at very low relative abundance. So this is really important in informing about how we think about the gut microbiota and how we design our experiments. So a lot of the diversity we retrieve might not be actually that biologically meaningful. And this is a key point about evenness. So evenness per individual is low. And what evenness means from an ecological perspective, it's about the relative distribution of species. So if a community is very even, so all the species are present at equal numbers. However, with the microbiota, you can see here that this is not the case. So we have 10 individuals here that are dominated by Saccharomyces. So you can just see those there. Saccharomyces is colored yellow. So it's typically present at 50% or higher in terms of relative abundance, but often takes over the entire community. So 100% almost of the sequences retrieved belong to Saccharomyces. You can see for Candida as well, this is in the pink. So it's highly prevalent, but it's prevalent at varying frequencies. So in some individuals, it's like 50% of the sequence reads, some it's less than 5%. Unknown fungi in the light green in the middle of the graph here, they dominated one individual, and we also frequently recover unknown fungi in other, but at much lower abundance. Another important point, and this will be talked about later, I'm sure, is that many food-associated genera detected using culture-independent analysis were present in our data sets. So we recovered Penicillium and, as I said earlier, Saccharomyces. So Saccharomyces, we did not actually recover this on our plates. So we also plated these samples on a range of different fungal media, and we did not recover it. However, we largely recovered Candida on plates. So this tells us something about Saccharomyces, that it's largely probably of food origin and it cannot grow at 37 degrees. So this moves on, I guess, to a key point that we need to think about in studying the microbiota. So how do we identify, I guess, what you would call functionally relevant fungi? So fungi that may be having an effect on the host or that are important, for example, for ecosystem stability and function. So a couple of the key points are that, as I said, most fungal diversity reported in the gut microbiota surveys, they are study specific. So we cannot generalize to large groups if the vast majority of diversity we recover is just present in an individual in one study. But also this diversity reported, these are usually present at very low relative abundance. And as I said, fungi of food and environmental origin are also commonly reported. So the point here is that it is likely that a lot of the fungal diversity reported are species that are actually not true gut colonizers. So the key question then is how much of the fungal diversity that we detect in these types of studies is actually biologically relevant? And this extends also to how do we actually go about then identifying biologically relevant fungi? So one thing we could do is we could focus our efforts on understanding the relevance of core mycobiota. So these are species that are commonly found across different cohorts, and commonly reported genera include Candida, Saccharomyces, Malassezii, Penicillium, Aspergillus, Deborah-Myces, and so on. Or should we just focus on growing species that we know can grow at the appropriate human physiological conditions? So those that we know can go, for example, in the presence of boiled salt at 37 degrees and so on. And here I think longitudinal sampling would be really useful to identify stable and colonizing species in this respect. But a key point as well is that something doesn't have to be colonizing to affect the host. So non-biological agents, for example, pollen and so on, medicines exert physiological effects on the host. And we also know that transient and non-colonizing fungi can affect the host. So Saccharomyces boulardii, which has been used for many decades as a probiotic to treat a range of conditions, related to acute diarrhea and so on. This does not colonize the host, but can actually have a profound effect on the host. So it's not just about colonizing fungi as well. Non-colonizers could also have an effect. So in terms of functionality then, what are what is the microbiota or members of the microbiota doing in the context of host health and disease? So I'm just going to talk about this in very general terms because, as I said, I'm aware that the other speakers might get into this in more detail. So from a beneficial perspective, what role might fungi be playing? So fungi, they're present in the neonate gut, so we acquire them at birth from either our mother or the environment. And do they actually play a role in developing a competent immunity? So we could have so-called probiotic fungi. So as I said, Saccharomyces boulardii has been widely used for decades to treat a range of conditions. And many fermented foods that have purported health benefits also contain a diversity of different fungal species. They're also a source of nutrients and vitamins. So for example, Saccharomyces cerevisiae, so brewer's yeast, has been widely used for a long, long time as a source of B complex vitamins and a source of nutrients and vitamins such as chromium and selenium and so on. So of course, then, it's a lot easier, I guess, to talk about potentially the negative roles of fungi in human health. And this is because many fungal species, and this is just generally, are well-recognized pathogens and opportunistic pathogens of humans, but also, of course, a whole range of other species. And there's a whole array of papers that have profiled fungal diversity in disease cohorts, and all of them have showed variation between case and control. And as I said, a key point here is actually trying to understand the biological relevance of that variation is the key question. And these are just a couple of titles that I picked out, just showing, it's been linked with IBD, colorectal cancer, and other conditions as well. But as we know, gastrointestinal disease is, extremely complex. The etiologies and the underlying, we'll say, mechanisms that are driving the disease remain unknown, and there's often a chicken and an egg scenario with the microbiota and gastrointestinal disease. So we don't know what comes first. Is it the disease state, and then you have particular bacteria or fungi that are well adapted to that state and proliferate, or are they actually driving the disease process? So causal roles for the microbiota are very hard to pin down, and that is true for the fungi as well. However, we do know that different Candida species, these are well-recognized opportunistic pathogens, and in vitro and in vivo studies have looked at Candida in particularly IBD, and this is actually a very active area of research, and I know one of the later speakers does a lot of work in this area. Also, fungemia is quite common, so this is blood infections caused by fungi, and I think Candida has quite a high fatality rate and is a very common source of blood infections. So then, just from an ecological perspective, what are they doing in terms of interacting with other, components of the human gut microbiota? So they're likely to be involved, there's likely to be fungal interactions and interactions between fungi and bacteria. And these are likely to be very important to ecosystem assembly, structure and function, and also disease processes. So I've just highlighted this here. So there's a really excellent paper out Coite that I worked with before is involved in this, and it shows how Candida plays a really important role in ecosystem assembly in preterm infants. And there's also a really nice paper out on bacterial fungal interactions in Crohn's disease as well. So there is precedent out there for interactions between bacteria and fungi, and also fungi in driving disease processes and in driving the ecology of the human gut. So just to finish with a couple of perspectives and a summary. So obviously this is an ISAPP meeting, so just from the probiotic perspective. So obviously fungal species have been used as probiotics. So Saccharomyces boulardii has been used for many years as a probiotic, and you know, without major side effects. Also, I think there should be a development at least, or a move towards developing probiotics to target specific fungi such as Candida. This could be one potential area of research. And as we know, yeasts are really important to synthetic biology. And this really excellent paper came out recently. It's about an engineered probiotic, so Saccharomyces cerevisiae, that could sense inflammation in the human gut and actually then respond by treating it. So this is just about 3 days old, and it's a really excellent paper on the utility of yeast as probiotics, of engineered yeast, I should say, as probiotics in, for example, IBD, and that was out in Nature Medicine. So to summarize then, so a diversity of fungi have been associated with the human gut, but much of this diversity that we see in the literature is unique to specific studies, and they're present at very low abundance. The diversity of the microbiota is uneven. At the individual level, we typically see microbiomes that are dominated by a single or a small number of genera. However, some genera are common across hosts and are common across datasets. So there does seem to be a trend emerging for a potential core microbiota. However, there is variation in the microbiota, and we see this across age, gender, diet, and disease. So understanding the relevance of this variation, I guess, is the key crux of microbiota studies. And so what we need to do is focus on identifying biologically relevant members of the microbiota. And that's a key area. Also, fungi, we know, can interact with bacterial members of the community, and they can also have positive or negative effects on the hosts. And just to finish then, Saccharomyces boulardii, we know it's been commonly used as a probiotic, but engineering strains of yeast for drug delivery and nutrient delivery as well is a real area that is gaining a lot of traction as well. So just to finish with a couple of thank yous. So Chloe Hussein was an excellent PhD student who did much of the work on these fungal projects. Professor Paul Cotter, who's a collaborator on these projects as well. APC, Royal Society, SFI for funding, Mary Ellen Sanders, and Eamonn Quigley as well, and ISAPP, of course, for the kind invite to speak today. And they're just my contact details if anyone wants to get in touch. So thank you again, and thank you for the kind invite.
And thank you, Pauline. That was an outstanding presentation, as you will see from the messages we're getting, and a great way to start the meeting. I should have mentioned at the outset that you're a Royal Society SFI Research Fellow at APC, having previously been a Marie Curie Fellow at Tagus in Moore Park, and many other distinctions along the way. That was fantastic. There actually are a lot of questions coming in, and I think Christine and Mary Ellen, we're suggesting that something we didn't want to suggest, namely that they could be answered online. Is that correct?
Yeah, I think the panelists are free to go ahead and respond to some of those if they would like, but we're going to save the verbal questions until the end, Eamonn. Hopefully we'll have plenty of time for a Q&A.
So I'm going to go ahead and introduce the next speaker, which takes me to another place where I've spent some time, namely in Nebraska, but I was in Omaha, not in Lincoln. And it gives me great pleasure to introduce Dr. Heather Hallen-Adams, who's an assistant professor in the Food Science and Technology Department in the Institute of Agriculture and Natural Resources at the University of Nebraska in Lincoln. She's got a long-standing interest in gut fungi, and in particular in relation to diet, which is of great interest to me because my patients are obsessed with Candida diets and how they can cure all their ills. But I notice also that she keeps lizards and is a keen knitter. So I'm not sure how she will weave, forgive the pun, these into her presentation today. But her talk is going to be about characterizing gut microbiota from healthy adults, conventional versus vegetarian diets. Welcome, Heather. We're delighted to have you.
Thank you very much. And not to disappoint anyone, I'm not weaving lizards or knitting into this. I did actually sequence one sample from a lizard once, and it wasn't very exciting. So, but anyway, I'm assuming everyone can see my screen and hear me, and we're all good. Okay, so I'm going to talk largely about a few studies we've done looking at conventional versus vegetarian diets. Going to kind of broaden out from that a little bit, just to make some greater generalizations on, kind of build on what Pauline said about some of the fungi you can expect versus the ones you can't expect to see, and the ones that we can expect to play a role or not in the microbiome, and then kind of conclude with a little sample from our work of a fungus that could actually be a probiotic. So why are we interested in fungi and the diet? Diet plays a huge role in both the microbiome and more specifically the mycobiome, the fungal component. And this makes perfect sense. We're talking about gut organisms, at least in this context. There are microbiomes for other parts of the body, but these things are going to feed off of what we're feeding off of, so we can absolutely expect that diet is going to play a role. And just a few examples of this, very well-known paper from David's group in 2014, Diet Rapidly and Reproducibly Alters the Human Gut Microbiome. In that paper, they really reported that they had cohorts that they put on a plant-based diet and then alternated with an animal-based diet, so both meat and dairy. And they'd do each of these diets for 2 weeks, and they could just see with everything, bacteria and fungi, but especially the fungi, that shifting the diet, you almost immediately saw results in the microbiome. And this was fecal microbiome, and they could bounce it back and forth, and very reproducible, very strong effect. Going further, fungi, again, heavily influenced by diet. Tommy Achtung and colleagues working with samples from the Human Microbiome Project, healthy volunteers, they make the fairly dramatic claim that all fungi in the stool of healthy volunteers could be explained by their presence in oral and dietary sources. And in that case, the oral fungi being some of the Candida albicans. If the patients or the subjects brushed their teeth more often, they saw less Candida in the gut. They always saw it in the mouth. I wouldn't go so far as to say this is a universal, but definitely a lot, strong influence from the diet. And then Fears and colleagues, including Iliyan, who will be speaking later today, also bring up the point as to whether and to what extent gut and microbiota are symbionts versus environmental transients. So in our study, we had healthy humans on a conventional Western diet. We did not take dietary data on them. That was all the information they gave, conventional diet. 45 subjects and 1 to 2 samples per subject. And in this case, I was piggybacking off of another study, actually a dietary intervention study looking at resistant starches. So I just took the very first sample, the baseline, and then the final washout sample. Additionally, we recruited humans on a vegetarian diet, and again, no data other than self-proclaimed. This could be vegan, lacto-vegetarian, lacto-ovo-vegetarian, so there may or may not have been animal-based foods in this diet. Smaller cohort, 15 subjects, 16 samples, only one person gave 2 samples. And finally, we've also conducted literature reviews, in this case, looking at 38 additional papers that have species-level information and are looking at healthy humans over a little over a 100-year period. So conventional diet, you've seen a similar chart in Pauline's talk. Again, don't pay too much attention to it, just look at the pretty colors. The summary is with our 45 subjects, 69 samples, we identified 72 taxa to the species level. 38 of them we only saw once, one sample, never again. So a little more than half were singletons. And conventional diet, the average sample had 3.4 different species in it, with a range of 1 to 12. And no species was common to all samples. The yellow in this that we see pretty predominant, that's Candida tropicalis. In this particular group, that was dominant over Candida albicans, which we more commonly hear of. Again, as Pauline mentioned, there's a lot of study-specific things that we see. These individuals were in Nebraska and at Manhattan, Kansas, and maybe there's just some dietary or environmental reason that Candida tropicalis prevailed. So that's our conventional diet. We look at the vegetarians, and it becomes much more variable. We have fewer subjects. We have 57 taxa identified, 27 only identified once. So again, just about half are singletons. Much more diversity. So our average here is 12 as opposed to 3.4 species in the conventional diet. And the range was from 2 to 25. Again, no species common to all samples. And where it really gets interesting is when we start to really compare the two diets. So if we look at the top 5 genera in each, and the way this is laid out, we have the genus, the percentage in the diet under discussion. So Fusarium had 88% of the vegetarian samples had Fusarium. And then in gray, only 3% of the conventional diet samples had Fusarium. 81% of the vegetarian diet had Malassezia. That's a fairly common organism to come up in microbiome studies. Only 12% of conventional diet. Penicillium, 75% of the vegetarians, 1% of the conventional. Aspergillus, 68 and 6%. Candida, which we think of as being kind of our king gut microbe, 63% of the vegetarians, so still present in more than half of the samples, and then 84% of the conventional. And then conversely, we look at the conventional diet, Candida topping out. Our next most abundant organism, Geotrichum, which we didn't detect in the vegetarians, was present at 46%. So vegetarians, the abundant organisms, we're seeing at 60, 70, 80%. Conventional diet after Candida, it just becomes really variable, much lower percentages for the highly abundant organisms. And just a very different suite of organisms in both. Now, if we look at some of these names, Fusarium is a very large genus that tend to be plant pathogens. So grain crops, cereals, banana has a Fusarium, pumpkin has a Fusarium, tomato has a Fusarium. Generally, they're up to no good in those plants, but very, very abundant in agriculture. So you might be able to make an argument that vegetarians, if they're eating more plants, they're just picking up more Fusarium through that exposure. The same case might also be made for Aspergillus in particular, also a common plant pathogen. Penicillium can be a plant pathogen, also a food production organism in cheese. And these vegetarians, again, we didn't rule out people who ate dairy, just people who ate meat. Going down to the conventional diet, these are some of what we think of the more common gut microbes. I'll talk about them in a little bit more detail in the future slides, but it's harder to make a really strong dietary-based argument as to why a conventional diet person would have more cladosporium than vegetarians, and that might just be luck of the draw in the individual studies. So taking the bigger view, our studies combined, we had 97 taxa in 85 samples, 48, again, just about half were singletons. Only 14 were found in 10 samples or more. And that ranged from exactly 10 samples for Penicillium F. communi. There's a group of related penicillia, including some of the cheese penicillia that you can't tell apart by ITS sequence. and up to 57 samples for Candida tropicalis. Now, if we broaden our view and look at our studies plus the 38 other studies I mentioned, we go up to 630 taxa, 516 of which are only reported from one study. And I wasn't able to get sample level breakdown from this, so it might be multiple people in a sample, but again, very specific, very long tail. It's been shown over and over that you're not going to sample to saturation with fungi. You can with bacteria, you can plateau, but fungi, every new study, you're going to get something new and different. Only 27 taxa are reported from 5 studies or more. So if we look at the most commonly reported genera, we have Candida at the top, Saccharomyces second in the list, then Penicillium, Aspergillus, a tie between Cryptococcus and Malassezia, Galactomyces, that's another food-related yeast, and then a tie between Rhodotorula and Trichosporon. And most of these are yeasts. If we take it to species level, Candida albicans is the top, Saccharomyces cerevisiae, which cannot be readily distinguished from Saccharomyces boulardii, the probiotic Candida. In these studies, there's about a one base pair difference at IDS. Candida tropicalis, other Candidas, Glabrata, Parapsilosis. We get our first non-yeast. 33% of the studies have Cladosporium, another species complex. This is a well-documented environmental fungus. Common in the air. There are a series of studies on the mycobiomes of the Apollo astronauts, and they checked every nook and crevice and cranny of these people, including the fecal samples, and they reported Cladosporium in the Apollo astronauts returning from space. So it's there, but we think it's an environmental organism. It's been documented forming fungus balls in the lungs. It's in the air. Another Candida, food-related Penicillium, Malassezia, which tends to be associated with human skin, but there could be a interior microbiome species. Another Malassezia and Dibariomyces, which Pauline also mentioned as one of the more common fungi. Some more Candidas, and then tied at 15% of the studies, there's a handful of organisms. I've highlighted Agaricus bisporus in there. That's the mushroom of commerce. That's portobello, that's cremini, that's the mushroom you have on your pizza. That does not grow at 37 degrees. That does not do anything at 37 degrees. It is definitely a dietary organism. And if we look at some of these others, Dibariomyces hansenii does not grow at 37 degrees. Penicillium species commonly are psychotrophs. They're going to pretty much cease being able to do much of anything at about 30 degrees. So many of these they might be common, but we can attribute dietary exposure. So if we want to say what makes for a good gut fungus, if it's going to be resident, it should be able to grow at 37 and probably colonize the gut or at least the digestive tract. If you're looking at organisms where the primary niche is the gut or the digestive tract, you're really limited to Candida species. There are others that we can find all the time, but Saccharomyces, again, there have been feeding studies that show if you have a strict Saccharomyces-free diet, you ceased seeing Saccharomyces after a matter of days. But it is being constantly administered, so it may actually play a role in many people. You can have organisms that the primary niche is the microbiome, but maybe not necessarily the gut. So Malassezia species on the skin, a Trichosporon, often associated with skin as well, Cryptococcus. And then you might have long-term colonizers. So Saccharomyces and Cladosporium, where we think we have good evidence that they're environmental, but you have repeated administration, that those could be possibilities. Also yeasts in the Dipodascaceae, which would include Geotrichum, Galactomyces, saprochidi. Some of these are cheese organisms. Again, we tend to see them fairly commonly and maybe commonly enough to have an effect. For your non-resident fungi, we have the dietary ones. So Dibariomyces, Penicillium, that are associated with cheeses, Agaricus, the portobello mushroom. Generally, these are not growing at 37. You have your environmental exposure, and that could be airborne inhalants. That could be spores that land on food and are ingested, but aren't really necessarily dietary. And then there's some that are just crazy. So you have Ramularia, that's a leaf endophyte. It grows in the leaves of living plants. I've seen that on multiple times. A variety of wood decay basidiomycete fungi, so shelf fungi or conch fungi, we find them. And that has to be environmental. These things don't grow at 37, but they pop up. It's probably spore exposure. So with the allochthonous fungi, the dietary fungi may not persist, but they may still play a role. And this is getting back kind of to the idea of a probiotic. You don't necessarily, if you're taking a probiotic, you're administering it daily, but if you cease taking it, it might be washed out of your system. So there's a possibility for something that isn't native, isn't colonizing, to still do something. And my example is Dibariomyces hansenii. Again, this does not grow at 37, and presumed dietary origin, cheese, fermented meat, detected in 23% of studies. And our first study with the conventional diet showed a negative correlation with Candida species. Statistically significant. If you had Dibariomyces in your fecal sample, you generally did not have Candida tropicalis, Candida albicans, Candida parapsilosis, Candida orthopsilosis. And these are closely related organisms. Dibariomyces actually has a Candida name, Candida famata, but generally, if you saw it, you didn't see the other Candidas. So we looked at that in a little more detail. We obtained a number of cheeses with multiple samples per cheese over a period of a year and a half from just the local grocery stores. 79% of all cheeses tested had Dibariomyces hansenii present either on the rind or in the interior, and this was by culturing. We obtained them in pure culture. We tested 42% of our strains against Candida albicans and Candida tropicalis lab strains at differing temperatures and pH, and 23 of our D. hansenii strains had killer activity. And the killer factor in yeast is similar to bacteriocins in lactic acid bacteria. It's a toxin that kills generally closely related organisms. We believe it's involved in competition. And what was really interesting, I've hammered in the point that Dibariomyces doesn't grow at 37, it's not going to colonize the gut, but cheese colonized by a killer strain retains killer activity at 37 degrees over several days. And that's shown in this picture here. We have a block of cheese sitting on a seeded agar plate assay, and there's this clear halo around it, and all around in the background is Candida albicans. And the clearing just shows albicans is not growing next to the cheese. The blue is not the native color of this piece of Romano. That's from the media. We use methylene blue in this assay. But so we have a possible mechanism to getting a bioactive killer factor into the gut and eliminating Candida, at least transiently. And that's something that we are continuing to pursue at kind of a low level. I have about 50 or 60 samples from a feeding study with healthy volunteers looking at before and after a diet and during a diet of a cheese colonized with what we know to be a known killer Dibariomyces. And we've sequenced that once. There was a little difficulty with sequencing, so we're gearing up to redo it in a somewhat more controlled manner, and hopefully that data will be out in the not terribly distant future. So in summary, few fungi are likely to be active participants in a gut community. most or all of them are going to be yeasts, and then you have this long, long tail of incidental species. The sources of the fungi must be considered, and it still isn't. I'm still seeing papers that will say all of the patients under treatment showed a difference in the microbiome from the uncontrolled or untreated patients, and then you read and you see that the treated patients were being treated in the same hospital on the same diet, and maybe that makes a difference. But people are becoming more aware of this. The environmental fungi don't necessarily have to be ruled out. They could still play a role, and that's based on the capacity of the fungus. Does it make an interesting bioactive compound like the killer toxin Debaryomyces? Does it make an antibiotic? Is it utilizing sugars that gut bacteria are making, and so on? It also depends heavily on the host and the host microbiome status, if the fungus is going to play a role or not. What I think we really need at this point is studies on gut fungal ecology, showing really what these things are actually doing. Open questions are the persistence of gut fungi, different niches, different species, different strains, whether the Dipodascaceae yeasts are strictly dietary or whether there is a more gut-associated one, similar with Saccharomyces, Malassezia, and Cladosporium. And with that, I'd like to acknowledge some former lab members who were involved in most of this work, Mallory and Nabaraj and Raisa, and all the graduate students under me. And with that, I will conclude.
Thank you very much, Heather. That was great.
So our next speaker is Professor Joseph Petrosino. He is the chair of the Department of Molecular Virology and Microbiology and the director of the Alkeck Center for Metagenomics and Microbiome Research at Baylor College of Medicine. And we're just very, very happy to have you with us today. Joseph, looking forward to your talk.
Great. Thank you so much for inviting me. It's great to present in front of this audience. Can you see and hear me okay? Good deal.
Yeah.
All right. So, advance my own slides. So I'll make a quick overview of what we're going to talk about today. So I'm going to look at the role of the microbiome and what we see as translational microbiome research in precision medicine. So translational science is obviously a very hot topic both in academia and industry, and then precision medicine, the idea of tailoring the ability to treat and detect disease and wellness in individuals in a way that encompasses all of us instead of a subset of the population. Go over a couple of sequencing technologies and strategies that we use to monitor the microbiome as well as other microbial components in the microbiome. And then we'll take a quick look, and this should say mycobiome in the HMP cohort, but the Human Microbiome Project was one of the few projects in 2005-ish timeline where the renaissance in microbiome research was kicking off thanks to the next generation sequencing technologies that were being applied to take a census of microbial communities. And so I'll talk about how that program that initially started in bacterial research, then we used samples and data to look at the fungal compartments in that cohort. And then I'll focus finally on a prospective cohort looking at type 1 diabetes and fungal associations, not necessarily just with type 1 diabetes, but also in a unique birth cohort looking at the evolution of the colonization of the fungal compartment of the microbiome in kids from 3 months to 4 years of age. So I won't spend a lot of time on this. This was alluded to in earlier talks, but we use a variety of technologies to extract and to design, sample, extract, sequence, and analyze microbial communities. It's important to know that when you're studying the microbiome, that things that work with the microbiome or the bacterial compartment of the microbiome don't always work. And so extraction methodologies, for example, have to be validated. And as you heard before, PCR primers are specially designed for fungi compared to bacteria, so you can hone in on those organisms specifically. We were here in Houston in the Texas Medical Center, so just across the street from Eamonn. One of the things that how we approach microbiome research is to approach these projects at scale. In other words, we like to look at multiple cohorts as much as possible for any study that we're working on or any disease that we're working on, because we realize that there are universal factors, things in common with type 1 diabetes, for example, at different corners of the world, where we can find universal factors, common features, common risks and exposures that can be used for diagnosis and treatment of disease. But we also recognize the fact that there are unique regional factors having to do with either host genetic or human genetics and host genetics, or diet or other healthcare practices or the environment itself that may give individual readouts that are not common to all people experiencing the same disease. And having a grip on both of those helps us design precision medicine-based paradigms that will hopefully allow us to be able to develop treatments that are targeted for people all over instead of just one location or one ethnicity and so on. So I won't spend a lot of time on this, but happy to discuss a little bit later. So here at Baylor College of Medicine, we've established a Center for Microbiome Research in 2011, stemming from our activities with the Human Microbiome Project in 2005. And the idea was to be able to coordinate and collaborate with individuals from all over the world to build these multi-cohort projects in a variety of diseases. Some of them are listed here, but being a highly collaborative center with capacity to sequence and analyze at scale, and recruiting investigators to be able to dissect the associations we find with our omics-based processes. So I'm going to talk a lot about associative studies in my talk, but you need to be able to do the functional studies that were alluded to and discussed in prior talks to, again, be able to make sense of what these associations really are. So with those tools here, we've engaged in a number of projects. And again, one of them involves studying the microbiome of the Human Microbiome Project Healthy Cohort. And so with just, without going into too much detail about the cohort itself, because it's been, I'm sure everyone's seen presentations about it and can find it easily in the literature. What we did is we took a subset of samples, gut samples from that HMP cohort that I'll talk about in a moment. And Andrea Nash, who's a graduate student at the time in my laboratory, and Tommy Akhtan, who is a faculty member now at the University of Nebraska at Lincoln, worked on these data looking at the fungal compartment of the HMP cohort, which was largely ignored in the initial analysis of HMP samples. So originally, 300 individuals were recruited to participate in a healthy cohort build out for HMP. This was done in between 2005 and 2009. 150 people from Baylor College of Medicine, 150 people from Washington University, St. Louis. And some of those individuals were sampled more than once. And here at Baylor, we had a subset of 50 people who were sampled 3 times over a course of 1 year. And we had extracted the DNA using a mobile power cell kit at the time. That kit has now been bought by QIAGEN, or produced by QIAGEN now, its latest generation. We validated that the power cell kit did a good job of extracting fungal members of the microbiome, and then we used those samples, that DNA, to amplify with ITS2 primers to look at what fungi we could detect in those samples and how that changed over time. One of the basic comparisons we did initially was compare the diversity in the gut fungi compared to those to the bacterial compartment. And so observed OTUs, operational taxonomic units, are shown here on the left, just from the first sample for each of the 50 individuals I mentioned. And you can see, as was alluded to before, the diversity in the fungal compartment is much lower than compared to the bacterial members. So OTUs are, again, not necessarily species, but you can kind of think of them as individual taxa within the sample that was collected. Shannon diversity introduces the concept of evenness into the also the richness of the community, and it too also shows that the bacterial compartment is more diverse than the fungal compartment. We looked at the abundance of taxa that were present of the top. We looked at the top taxa that were present in the fungal yeast compartments, and found that these were the top. I believe this is top 15 present in the samples that were collected. We heard some of this data alluded to before, or some of these observations alluded to before in other cohorts, but just to follow along the same theme: Saccharomyces, Malassezia, Candida, Cyberlindera, and Penicillium were among the top 5. Some are yeast, some are molds. A lot of them are introduced by other body sites or our environment or food sources. One of the things that we wanted to look at was how well intra and intravolunteer fungal diversity was. So in other words, if you look at a sample at a given point in time, how likely is it that the second sample you take resembles that first sample? So so it's known in bacteria that between volunteers diversity, there's changes when you look at the microbiome between 2 individuals, and that's much more different than looking within an individual. So your own sample and then collect it again. So you're much more like yourself than you are somebody else. With the fungal compartment, that's not necessarily the case. So we see that the difference from a person's sample 1, sample 2 is about as different as a person's sample compared to another person's sample. And so shows that the fungal diversity, inter-volunteer diversity is quite high. And then within volunteer diversity, I mean, it's quite high. We want to look at the correlations between fungal and bacterial compartments, what fungi either promote or restrict the growth of other bacterial components. And we also did this with fungi, but I'm not going to go into them right now. So in this case, we saw that, for example, Saccharomyces and Penicillium had negative impacts on the growth of Lactobacillus and Faecalibacterium. Similarly, Cladosporium and Ruminococcaiaceae also had a negative correlation to each other. And why do these studies? It's one of the goals of microbiome research and developing new therapies for building healthy communities for wellness or modifying a community for treatment of certain diseases. It gives us other opportunities and potential tools to administer organisms that could reduce or encourage the growth of members of the bacterial or fungal compartment going forward. And so these things could be actually turned into drugs in the future. And so in contrast, for example, we saw that botrytis and Rechinelliaceae were positively correlated. So if you want to influence the growth of one taxa, you introduce the other is the hypothesis that you would have to test with this. So focusing a little bit now on a different cohort that's really amazing. You'll understand why in a moment. Gives us a really interesting insight on the development of the microbiome in infants to toddler age children. And so this is the TEDI cohort, and TEDI stands for the Environmental Determinants of Diabetes in the Young. It's a prospective cohort of children that are predisposed to type 1 diabetes based on genetic profiling of HLA, of the HLA genes. Also, if you have a first-degree relative that has type 1 diabetes, you could be enrolled as an infant through your family into this cohort. And the families are really special. The parents are writing down diary entries, about 1,300 metadata points per month. So psychosocial stressors, family habits, things of that nature, and samples being collected from the babies on a routine basis as well. So stool samples that are monthly through age 4, blood samples quarterly, many other types of samples. And so we set out on a project looking at the bacteria, the fungi, and the viruses associated with this cohort, looking obviously for associations with autoimmunity and type 1 diabetes, but also stepping back and saying, there's 8,600 children in this cohort, and we have some monthly samples from them. Can this give us a unique insight into the developing microbiome in these children? So, and we're working with a large number of investigators. I had them on the last slide, but for the sake of time, won't go back. Large number of investigators throughout a network of 6 clinical centers. So Sweden, Finland, Germany, Seattle, Denver, and groups in Florida and Georgia, all participated in recruitment and sample collection for this program. So you also can get geographic information about the cohort and the microbiome as it's developing. There are a couple papers that started to look at the characterization of the bacterial compartments that were published in 2018. But Tommy, when he was here, and actually now also still while he's in Lincoln, has also been looking at the fungal compartment from the data set that we collected from these children. Just to kind of give you a point of reference from the bacterial side of things, we found that there were 11 distinct or 10 distinct communities that were found, bacterial communities found in the guts of children over time, and they became more diverse over time. We found that breastfeeding, furry pets, the presence of siblings, and antibiotic use all had a dramatic impact on the statistically significant impact on the 903 children and their accumulation of microbes from birth to about 2 years of age. And in this case, we did find that there were viral associations with higher risk for type 1 diabetes. So enteroviruses, in particular enterovirus B, family of viruses, having longer exposures to those viruses were associated with children having increased risk for getting type 1 diabetes. And interestingly, there was an observation that adenovirus C exposure within 6 months of birth also seemed to have a protective phenotype looking at type 1 diabetes onset. So this is all published, and you can go look at that if you're interested. But we, again, wanted to look a little bit more at the fungi in these children. And so that's where the next several slides are going to address. So we looked at initially, again, over time, just the abundance of fungal reads in metagenomic samples. So the relative abundance of fungi that were present, and showed that we do indeed see accumulation of fungi from birth to about 2 years of age. And then after 2 years of age, they level off regardless of geographic location. There doesn't seem to be necessarily a geographic, there's no bias in terms of which area of the world you live in terms of the fungi that you accumulate. And as related to the HMP cohort or a cohort of non-Western adults, so more from pristine cultures and other studies that have been done on the microbiome, you see that the infant and the child microbiome are a little bit less diverse, at least in adults. So there's still room to grow, but approaching that of non-Western adults. Looking at ITS2 data from 9,500 samples across 825 children, you can see a little bit more direct the diversity comparison between fungi and bacteria in these infants. As you expect, the bacterial membership diversity grows over time in the first 2 years of life and then plateaus, whereas fungi pretty much stays the same. So while fungi are accumulating in terms of load, and that's what we were looking at the last slide, the diversity isn't really changing too much. So the taxa that are present are staying relatively even throughout the first couple of years of life. We did the intra versus inter-volunteer comparisons and showed that so this is becoming more, this is Bray-Curtis similarity. So as you go up the y-axis, you become, your two samples are more alike. And if you stay closer to the x-axis, they're more different. And so going from top to bottom, you see that within volunteer bacteria has the greatest sample-to-sample likeness or similarity. Then the fungal compartment, then fungi. So in this case, the fungi in these infants, two samples within the same individual were the next most similar. And then bacteria between volunteers, and then fungi between volunteers. And that's the profile that we saw across 48 months. The 5 most abundant taxa appearing over 3 to 48 months are shown here. And so obviously the most dramatic increase is in Saccharomyces cerevisiae. If you look at the diet of these children, you can get a pretty good impact or idea of why Saccharomyces is appearing, and that is it's the introduction of food, so solid food. And meanwhile, we still see penicillium dropping significantly over that period of time, although starting to come up a little bit towards the end. And the presence of 3 different Candida species. Comparing HMP to samples in TEDI, we want to look at what were the carriage of organisms that were the most abundant in each of these cohorts and relate them to the other. So in this case, looking at the taxa that were the most abundant in TEDI, listed here again from the last slide, we see that they're present at different levels within the HMP healthy adults, shown in this brownish red color. So Saccharomyces cerevisiae were high in both cohorts, but you can see the difference in penicillium Candida in these 2 Candida species in particular adults, children.
Hey, Joe, just want to let you know you've got about 2 minutes.
Sounds good. Relative abundance and select taxa, these were the ones that were most different overall between the two cohorts. And you can see that this gives you an idea of what the difference is between the taxa that are different between children and adults. And while the samples are collapsed here, these actually average out in most of the samples to be near zero in terms of percent reads relative abundance. Impacts on the microbiome over time, breastfeeding. So there's a little bit of difference, especially in, I think this is, yeah. So especially in the first 18 months of life, you can see that there's a difference in the amount of fungi that are present based on whether or not you're breastfeeding, based on whether or not you're taking probiotics. Children that have asthma, that go on to have asthma, actually seem to have a different carriage of fungi. And then, interestingly, there's a gender difference observed as well. And when you have this many samples, you can see sometimes these subtle differences start to play off. We'll need to dive into more what this necessarily means. Geographic location has an impact, and for sake of time, I won't go into them too deeply. There's no apparent difference. The big question is in terms of type 1 diabetes. There's been papers associated with Candida albicans as being associated with autoimmunity in type 1 diabetics. We didn't see that in this cohort. That doesn't mean it doesn't exist. It's just the way we've looked at things at this point in time. This strain, this fungi was associated with asthma in a study of Ecuadorian children, 27 Ecuadorian children previously. We did not see it associated with asthma. In fact, we saw a reverse association with it, potentially in terms of a trend. We saw that Candida Saki actually had an association with children who went on to have celiac disease, which is there's a large prominence of celiac cases within the TEDdy cohort, so it gives you an interesting sub-study that can be done with these data. Candida albicans did not have a similar association, so this also is something that's being selected for further study. So this tells you everything I just showed you, so I won't take more time on doing that, but I think the bottom point is the one that's most important, is that we have to follow up with mechanistic studies on these observations, we and others, as there's a lot of data to follow up on. So with that, I want to thank Tommy specifically in all the work that he's done with the TEDdy cohort, and Tommy and Andrea with the HMP cohort. And with that, thank you for your attention.
Thank you so much, Joe. I really appreciate your talk about this cohort. So next, what I'd like to do is introduce Professor Eamonn Quigley. You've seen him already today. He is giving the next presentation on the implications from a clinical perspective of the mycobiome. Professor Quigley is at the Department of Medicine, the Academic Institute Director, the Linda K. and David M. Underwood Center for Digestive Disorders at the Houston Methodist Weill Cornell Medical College in Houston, Texas. Now, Eamonn was a little bit worried that he would have a clinical conflict with what was going on. So he did pre-record his talk, but I'm going to go ahead and share that with you.
I feel a little bit awkward being positioned in this symposium in the midst of real scientists, but what I'm going to try and do is provide a clinical perspective. And my clinical perspective is a very simple one. It is this. What I contend with on an almost daily basis, patients who arrive telling me they've got candida, and that this candida is responsible for various symptoms. And if you look online at these various sites that people watch, you think candida is almost an epidemic. And this is an example of one of these sites. And you can see here a myriad of various complaints which have been associated with candida infection. Some of them genuine, like fungal infections of the skin or nails, though here it's also debatable. It could be another fungus. But then there are lots of other things like autoimmune diseases, difficulty concentrating, poor memory. The list just goes on. Not only do you have these claims of all sorts of associations with candida infection, but there are also, of course, remedies. Diets and cookbooks that can cure all of this. And here you have one site which is telling you the strongest candida killing foods. However, all is not despair when you come to look at the internet, because here's a site that I really liked that asks the question, is gut candida overgrowth actually real, and do candida diets work? And there's this beautiful summary here. They say how nutrition scams work. Take a kernel of truth, blow it up using fear, misinformation, and false claims, convince people they have a scary problem that has vague symptoms that they can't be tested for, sell a cure for the problem, usually involving a special diet and supplements, and convince people that they're better because of this specific cure. And that summarizes a lot of what I've got to put up with. So, having said that, of course, there are instances where fungal infections are important in the GI tract. For example, patients who are taking oral or inhaled steroids develop candidiasis of the oropharynx and esophagus, and we see this all too commonly in patients taking steroids for eosinophilic esophagitis. And then, of course, there's the immunosuppressed patient, such as the patient with HIV who's on chemotherapy, transplant patients, and especially patients who've had small bowel transplants, and those who are immunosuppressed for other reasons, such as liver disease or malnutrition, or who have an inborn error leading to immunodeficiency. All of these are prone to fungal infections, which can be devastating. Indeed, this very nice recent paper summarized the risk factors for candidemia, a severe infection, such as neutropenia, having a solid organ transplant, having significant liver, respiratory, or cardiovascular disease, recent gastrointestinal, biliary, or urological surgery, central venous access device in place, intravenous drug use, having urinary catheter in situ, and being treated with carbapenem. Nobody would dispute the role of candida or other fungi in this situation. And then a few years ago, there was this paper, which is an important paper, which brought to our attention the issue of candida diarrhea. Remember now, this is a small series of just 10 patients with severe diarrhea. They were all elderly, malnourished, and critically ill, suffered from chronic debilitating illnesses. They've been treated with multiple antibiotics and chemotherapeutic agents. They were very sick with dehydration, renal failure, electrolyte imbalance, stool cultured most commonly as Candida albicans. There was no evidence of colitis, and the diagnosis was based on the continuation of diarrhea despite fasting, the exclusion of other causes of secondary diarrhea, and a dramatic response to a short course of nystatin. But this, I emphasize, is a rare scenario. Now we come to some of the areas that I have more problems with. And here's this concept of small intestinal fungal overgrowth. So here they took 150 patients with unexplained GI symptoms. They aspirated fluid from the duodenum, and they cultured for aerobic and anaerobic bacteria and for fungi. You can see their definition of bacterial overgrowth and their definition of fungal overgrowth, the latter being just producing any fungal organisms. And they also assess motility. And what they found in these patients who probably had functional GI disorders, that 63% either had bacterial overgrowth or fungal overgrowth by their definition. Some had both. And as you can see here, somewhat interestingly, dysmotility was a risk factor for both bacterial overgrowth, regardless of criterion that you used, and for fungal overgrowth. But being on a PPI actually seemed to have contrary effects. It made bacterial overgrowth more common, but seemed to be less common among those with fungal overgrowth. So I'm not sure what to make of this. But critically, this particular study did not assess the response to treatment, so we really cannot conclude that fungal overgrowth had anything to do with these patients' symptoms. More recently, my former colleagues in Cork looked at the microbiome with irritable bowel syndrome, and as you can see here, saw no differences in richness or in diversity, either in IBS patients as a whole compared to controls or between different subgroups of IBS patients. And this shows you the distribution of fungal organisms that they isolated. And you can see here that there are some differences between control and IBS, but the important point to make is that Candida, Saccharomyces were commonly found in both controls and IBS subjects. So here's a summary of their findings. They found the beta diversity differed between IBS and controls. The microbiome covariated with the bacteriome and metabolome. The microbiome was not linked to dietary habits, very importantly, and the predictive power of the microbiome was better than random, but was insufficient for clinical diagnosis, really disputing a strong association with the microbiome and functional symptoms. This is another paper which looked at normal volunteers and patients with IBS who were subdivided according to whether they did or did not have visceral hypersensitivity. Both groups had a difference in Shannon diversity compared to healthy volunteers, in contrast to the paper from Cork. And you can see here the distribution of fungal organisms, again showing some differences, but again more or less the same distribution of fungal species in the two groups. They went on to show that this might be relevant to visceral hypersensitivity in an animal model, even though in the human studies there were no difference between those who did and did not have visceral hypersensitivity. Finally, and I'm sure we're going to hear more about this today, this is from Dr. Petrosino's group, showing the effects of diet and teeth brushing on the microbiome, and showing here that if you took on a regular diet, you isolate Saccharomyces. If you go on a Saccharomyces-free diet, you virtually eliminate them. Then just have one bite of bread, they reappear, then disappear, only to reappear again. And shown here down on the right is the effect of teeth brushing on the red dots when you brush just once a day, in the green dots when you brush 3 times a day, and you can see the dramatic differences in the isolation of Candida albicans. So in summary, and again, this is my perspective, the blogosphere, unfortunately, is rife with theories on fungal diseases, prevention and treatment, and they provide a starring role for Candida albicans, the source of all ills in the world. Data on the human microbiome is limited to a few studies on fecal samples. Though the concept of small intestinal fungal overgrowth has been postulated, its clinical significance remains to be defined. Yes, you can detect fungi, but you can detect fungi in normal individuals. But without a treatment success, we really can't claim any role in symptoms. I would address this to all of my colleagues today. We need more data on normal fungal communities in the entire GI tract, in health, in disease, in relation to diet. And I would ask them, are there new technologies that may help the clinician who is currently lost? And with that, I thank you for your attention and look forward to your questions.
Okay, thank you so much, Eamonn, for that. And now we're going to go directly to the last very brief talk. We have a 5-minute presentation to give us a bit of an industry perspective on the microbiome. And joining us to provide that is Dr. Frank Schuren from TNO Netherlands Organization of Applied Scientific Research in The Hague. And he's with the Department of Microbiology and Systems Biology.
Okay, I hope you can hear me well now. Okay, thank you. So I will start off for this short presentation. I think we heard a lot of very interesting data today about this field of the microbiome in relation to human health and disease. Mainly targeted to the gut situation, but I think it's also important to mention that it's different body sites where fungi do appear, and both in relation to a healthy and a dysbiotic stage. One of the things I feel which is still open in the field is whether, and that may differ between these different niches in the human body, whether there is really a stable, healthy microbiota situation. But I think we will learn that with continuing research. So just to mention one other area which is really still very much open, but which is not discussed today, it's fungi in relation to neurodegenerative diseases. There's a couple of papers in the scientific literature where it was shown that people which have different types of neurodegenerative diseases, that the presence of fungi could clearly be presented. And of course, the chicken and egg discussion applies here. So no one knows what's cause and what's effect. But there's potential that fungi play a role with many more diseases than currently looked at. So looking more from an industry perspective and different options, I think it's a couple of areas which are of interest. One, it's diagnostics. I think the main focus today was on molecular diagnostics, which is certainly very powerful. Of course, the more traditional diagnostics by culturing organisms, certainly an option, but there's also room for novel diagnostic approaches in humans, especially if you're looking at fungi in different body sites where a lot of the current options are not readily feasible. So you could think about detecting cell wall components, for example, which may end up in the blood, for example. Then we move on to the prevention area, that which, especially for the industry partners involved in this discussion, it's probably one of the most likely areas. I think it's different elements which can be of interest. One could look at really targeting fungi with antifungal treatments, which could either be targeting specific fungi or fungi more in general. Looking at all the presentations today, it's very difficult to pinpoint one specific fungal species. So maybe the broader approaches could be more of interest. This might include microbiome modulation, competing with fungi, could also include novel type of prebiotics by stimulating bacteria in such a way that fungi get problems, that, but at the same time could also include probiotic approaches. And I would, for my personal feeling, this prevention approach might also apply to early stages of disease development. So we should be able to identify disease development in early stages where, in principle, the same approaches might still apply. And at the same time, individual variation is very important, as also mentioned many times today. So this is certainly something to take into account. Well, one approach we are working on, it's mimicking situations as they appear in humans, but then in experimental setting by developing, for example, microbiome screening platforms where you also can include fungi, which would allow for screening all kinds of components, prebiotics, probiotics, other type of ingredients that before going into animal or human studies. Well, and then, of course, as a follow-up, like in all cases, you need to go into human studies. But this would also need specific approaches, taking this individual variation into account, which is also in this field, like in the whole microbiome field, an important variable. But there's certainly ideas how this could work out. Well, then to summarize, I think what's shown today, that microbiome in human health and disease is a very interesting new field. There's many novel developments, both presented today, and also if you look into scientific literature, there's a lot of activity ongoing. As usual, more research is needed. And probably the currently most likely industry options would be prevention of fungi-involved diseases, where you could work at early stage and target broader target groups, or go into more specific diseases where you would focus on early treatment. And with that, I would like to finish and thank everyone.
Thank you so much, Frank. I appreciate that. Well, now I'd like to welcome all of the speakers back to we'll go ahead and put everybody up on gallery view so everyone can see. I'm wondering if anyone has any pressing questions that they felt were asked, or should I just start with one of the ones that I've generated? Okay. Well, I'm just wondering if I could get a reaction from one or more of you on really what the take-home message is about gut fungi and health. And as Eamonn really alluded to during his presentation, there's just so much talk about problems with fungal overgrowth, and people are very convinced it's real. And there's talk about dietary sugar causing an increase in these in Canada and possibly other fungi. And I'm just wondering, what do you guys think about that, especially for maybe the average person or a general population member?
It makes it very frustrating to do a literature or especially a web search on Candida, as Eamonn so clearly pointed out. Most fungi, most of the time, are not doing harm. They're probably not doing anything in most cases, especially the environmental ones. Candida, yes, it definitely, there are multiple species of Candida. Many of them can be pathogens. They can do things. Most of the time, they're not. I mean, I think a big question that still is unresolved is, what is Candida doing in the healthy microbiome? And we know it is in the healthy microbiome. And probably, if it's there, getting rid of it just because it's Candida is not a good idea. It's deal with it when it becomes a problem. And we know the situations in which candidiasis risk is elevated. Again, Eamonn listed some of them. We know if you're going on long-term antibiotics, if you're having implanted medical devices, if you're immune suppressed, if the microbiome is disrupted, if the gut barrier is disrupted, those are all areas of concern. And otherwise, for the most part, probably your Candida is just happy to sit there and steal a little bit of the extra sugar that you're eating.
Anyone else want to weigh in on that?
I can add a bit actually on the, so there are some functional aspects. We have been looking into that for a long time, and that's what I went actually through in the last part of the presentation, but didn't go through all the aspects of that study. So basically, what that data is showing is that Candida, at least we detect those antibodies for Candida albicans, but some of them might be cross-reactive with other Candida species, but there is some level of education of the host immune system by actually the presence of those organisms. And this is important. At least we could actually see that in mice. I don't know how important it is in humans, but those antibodies exist in many individuals that we have been actually, healthy individuals that we have actually sampled. So in mice, if you actually target the B cells that are producing those antibodies, during immunosuppression, there is actually a higher susceptibility to fungal infection, and vice versa if you actually give back those antibodies with specificity or cross reactivity with Candida albicans, they're protective in that situation. So probably there might be some aspect of actually having Candida albicans in the healthy microbiome as an organism that educates host immunity, and that in some aspects protects us from fungal infections, because we don't actually suffer from those fungal infections if we don't have some very, detrimental mutations. And I was going to show that data in CAR9 in humans that we have and others have shown, but I didn't have time for that. So if that doesn't happen, or if somebody is not severely immunosuppressed, there might be this aspect of actually having those organisms as educators of our immune system. So that's a positive aspect.
Okay, fair enough. Anyone else? Pauline, did you want to say something? Yeah.
Well, I just think both comments just highlight the complexity of the situation, and it just underlies everything that I guess the microbiota we have and the effect it has on us. It's a context-dependent process. So for the vast majority of people, exposure to fungi is probably, important to educating our immune system and so on. But if you are severely compromised, the fungal microbiota, the microbiome generally, the context is different. So in those scenarios that Eamonn outlined, that Heather and Iliyan have spoken about, that's, that's when they may be problematic. But generally, I think most people, it's not something they have to worry about.
We had an interesting question more from an environmental point of view. The question was, with soil health in conventional agricultural declining, would you expect to see a decrease in diversity in the microbiome compared to that of someone who might either be eating, more organic or closer to the earth type diets versus ones that are more maybe industrial type diets? Helen?
Probably not really necessarily. At least not based on soil health. There have definitely been things looking at organically produced vegetables and food versus conventionally produced, and we do see a decline in the food microbiome and in the soil-associated microbiome for the kinds of things I was looking at, where we see maybe more fusarium and aspergillus in vegetarians. That's not going to differ hugely between conventional and organic. And I think, at least at the level and scale I've looked at, I wouldn't expect that to be a factor I could really tease out without a really enormous data set, and then it would be very small differences.
Great. Well, Eamonn joined. I don't know, Eamonn, if you had any specific questions that you wanted to address the group with. Oh, you're on mute.
Yes. So this would be to all of you in terms of, for us as clinicians who are being asked these questions all the time by patients, what's the future hold in terms of making a diagnosis of when changes in the microbiome are maybe clinically significant as against being biologically important or physiologically interesting?
Take an initial stab at it. I think it's a great question and something we're all striving toward in the data that we're collecting and analyzing. I think it's going to be multifactorial. It's the sort of the systems biology of the individual. So collecting walking into your doctor's office and having him or her having your genome sequence and the predispositions associated with that are known. Having microbiome samples that are collected routinely, maybe annually, maybe more. Probably needing to use measures that are not the ones we're using right now in terms of stool collections all the time. But there are new ones being developed all the time. Just was reading about one where you swallow a pill that monitors gases throughout your digestive tract. So getting better analytics on the microbiome, and then, but being able to tie it to disease, environment, host genetics, and then having your diagnostics built off of that. That's what we're striving toward. And generally, we're not there yet.
I can add to that, indeed, in the same line. I think the field is following more or less the developments we've seen in the microbiome field, where for a long time there was also a struggle what this meant for human health in more detail. You see this field developing more and more and coming into specific applications. That I'm quite certain with the microbiome we will follow the same line, but we need more data and to look more specifically. And of course, we cannot copy the microbiome developments, but we can look at that and learn from that and relate the data which are being collected to patient phenotypes and probably have other readouts there as well.
Can I add one question, Pauline? You mentioned about the limitations of metagenomics at the moment in terms of application to the microbiome, which of course is the same technique has been an enormous aid in understanding the microbiome. So do you see progress in that area?
I think it's horses for courses, really, Eamonn. So I guess it depends on the question you're asking. So if fungal sequence, say if you just did a metagenomic prep on a DNA sample and 0.1% of that was fungal DNA, it's virtually impossible to assemble MAGs, like metagenomic assembled genomes from that. So in that sense, you will be doing an awful lot of heavy lifting to get very little. So, I mean, I've no doubt there will be developments in this area, but how useful they are generally and to the clinician, I think could be a lot of hard work for nothing, really. If you look at Candida, for example, and I think we should have kind of maybe a focus on maybe Candida as a model organism to better understand fungi and their role in human health. You can readily isolate fungi from a fecal sample. You can get count data on it. You can get genetic resolution down to the strain level. So there would be it would be a pointless enterprise, for example, to do a metagenomic sequence analysis, based on that rationale. So I think in terms of methods developments, I just think that culturing and sequencing, whole genome sequencing, and getting information on, we know that load, microbial load, like, you know, that's a key parameter in disease or infectious dose, you know, getting numbers and getting strain level information, I think will be really important as well. And coupled to what Joe spoke about earlier, you know, just real complex and detailed information is required from all angles, you know. But I think there's a lot of basic things that could be done a lot better.
But my problem is trying to explain that to somebody who's got Genova Diagnostics printout in front of them.
I know.
So-
I mean, it's not trivial for sure, and I think it's very early days. And
But I think you all did a wonderful job, and I wish we could
Keep
package
going.
that and tell the general public about it and get them to stop being worried about candida, which is their obsession.
Okay, so thank you so much. I think we're out of time now, and unfortunately we'll stop our discussion. I just want to thank all of the panelists. You guys did a great job. Thank you for really embracing this whole topic area so nicely and sharing your knowledge with so many attendees. And thank you also for the attendees and participating. Just to follow up, you will receive an email from Zoom, and there is a survey embedded in that. If you're willing to fill that out, that can give us some feedback on future webinars. And as I mentioned already, a recording of this with most of the talks will be available within a few days on the ISAPP website, isappscience.org. So thank you very much for your participation, and we'll call that a wrap. Thank you.
Thank you.
All the very best.